Hi students. So in the last session, we discussed about plant diseases, the general symptoms of bacterial, fungal and viral diseases, and about its control measure. So now let's see which are the main bacterial diseases. So the first one is Agrobacterium tumefaciens. It's a bacteria responsible for causing crown gall diseases in plants. Agrobacterium tumefaciens belong to the order Rhizobiales and phylum Proteobacteria and family Rhizobiaceae. The main symptom of crown gall disease is the formation of galls and large outgrowth on various plant parts including roots, and the growth develop mostly in the woody plant part. When they are young, galls are light colored and smooth, but as they mature, they become dark and woody and may crack and slow off. A crown gall is a solid mass of tissue in contrast to many insect galls that are hollow. Agrobacterium tumefaciens are gram-negative roots and they enter easily through the plant wounds. The optimal temperature of growth is 28 degrees Celsius and the ability to cause crown gall disease is associated with the presence of Ti plasmid within the bacterial cell. Ti means it's tumor-inducing plasmid. The tumor formation takes place in 3 stages. The first is the transformation period, where the normal host cell receives the tDNA from the pathogen. Now, the pathogen is the Agrobacterium tumefaciens. The second phase is the duplication period, when the tumor cells are changed by the bacteria due to the action of tDNA. And the third stage is the differentiation and organization period, when the plant hormone auxin start acting on the transformed cells. In the first step, the bacterium enters the plant through wounds and it becomes attached to the wounded plant cell. And then the bacteria enters stem or root via the wounded plant tissue and it multiply and spread intercellularly. And the bacterium also introduces a portion of its genetic material into the plant's genetic material, and this genetic transformation causes the plant to produce excessive hormones and compounds called opines. And these hormones cause the plant to overproduce new cells, forming a gall that the agrobacterium can live in. Opines are a special food that only agrobacterium can use. And this gall formation exerts a lot of pressure on surrounding tissue. And the tumors that is formed due to the transmission of tDNA, it becomes old and become woody and hard, and the young tumors are easily injured and it is attacked by the secondary invaders. Then the bacteria from gall surface move into the soil due to tumor breakage, and it can again infect other plants, and this process continues. Crown gall is nature's own genetic engineering. And the best way to manage crown gall is to avoid it. Do not purchase any plants with abnormal growth on the stem and avoid unnecessary wounding of plants, because it's through the wounds that the bacteria is getting entry. And if a plant in the landscape does develop crown gall, it should be removed. Otherwise, it can infect other plants also. And the next disease is bacterial blight of rice. Actually, rice is perhaps the most widely cultivated food crop world over. And this bacterial blight of rice is caused by Xanthomonas oryzae, and it is one of the oldest known diseases and was first noticed by the farmers of Japan. Xanthomonas belong to the phylum Proteobacteria and order Xanthomonadales, and it's a gram-negative bacteria. And this pathogen, that is Xanthomonas oryzae, is a yellow slime-producing motile gram-negative root with a polar flagellum and enters the host normally through wounds or natural opening. It reaches the vascular tissue, particularly the xylem, from where it multiplies and spreads throughout the plant. Symptoms are observed at the tillering stage. Tillering gives the crop the necessary number of stalks required for a good production, and the disease incidence increases with plant growth, peaking at the flowering stage. Prussic is the more destructive manifestation of the disease, wherein the leaves of the entire plant will turn pale yellow and wilt during the seedling to the early tilling stage, resulting in a partial or total crop failure. Yellow lesions are also observed on the leaf blades, and these lesions acquire a whitish straw color over a period of time. And also bacterial oocysts are also being observed in the infected leaves in warm and humid climates, and this will contribute to the spread of the disease. And the rice plants become infected with the Xanthomonas through rice seed, stem or roots that are left behind at harvest. Also, this leaves on dead plants and seeds and probably move plant to plant through water irrigation. And upon introduction to the host plant, the bacterium infiltrates the plant through natural openings, or it can enter it through root wounds. And Xanthomonas aureus grow in the plant and infect the plant leaf veins as well as the xylem, causing blockage and plant wilting. Bacteria oozes from leaf lesions and is spread by wind or rain, especially when strong storms occurs and cause wounds to plants. You can see that wilting nature of the rice plant. Management of bacterial leaf blight is most commonly done by planting disease-resistant rice plants. PSB RC82 is the standard variety of rice used in Southeast Asia. And the next bacterial disease is angular leaf spot of cotton. It is also known as bacterial blight of cotton. It is a disease affecting the cotton plant resulting from the infection by Xanthomonas citri pathowar malvesium. It is most important bacterial disease on cotton which infects all aerial parts of the host, and it is having a significant loss on different cultivars. The bacteria can infect the cotton plant during all growth stages, infecting stems, leaves, balls, causes seedling blight, leaf spot, black on stem and petioles. On the leaves, scattered small dark green water-soaked areolate spores measuring about 1 to 2 mm on the lower surface is also found. The spores increase in diameter to 5 mm and become angular due to leaf veination, and it becomes brown and later turn dark brown to blackish, becoming visible on the upper surface. Also, chlorosis, necrosis and distortion can also take place gradually. And the bacterial blight infection of cotton fields may begin from infected crop residues from a previous season or be introduced on an infected seed. The bacterium can survive in infested field debris and soil. Though the duration of survival is not well understood, it is likely that it is dependent upon the environmental conditions. So the infections may be spread by wind-driven rain from an infested source or during the time of irrigation, that is from the sprinkler and all. Also from the tools, tractors and other equipment that is used in the field may also result in the spread of this bacteria. If the plant is affected at the seedling stage, the bacterial blight will be more severe. The bacterium can enter the plant through openings such as tomatoes, lenticels, hydatodes and wounds when plants are damaged by wind-blown sand. And also the use of resistant cultivar is the most economical option to minimize yield losses from bacterial blight, and also practice aseptic techniques. The next disease is citrus canker, which is caused by Xanthomonas citri, and it affects the leaves, twigs and fruits of citrus plants, causing the leaves to drop and unripe fruit to fall to the ground. All the types of citrus plants are affected by this disease. Xanthomonas citri belongs to the phylum Proteobacteria, and order Xanthomonadales, and class Gamma Proteobacteria. Citrus canker is more severe in hot, wet areas. The infected sites ooze sap, which can carry the disease from tree to tree by irrigation or when rain splashes. Citrus canker can spread quickly over long distances on infected citrus roots and leaves, as well as on the people and equipment. There is no cure for the disease, so any infected trees have to be destroyed and replanted at great cost. The disease plants are characterized by the occurrence of conspicuous raised necrotic lesions that develop on leaves, twigs, and even the fruits. Epidermis may become ruptured by tissue hyperplasia induced by the pathogen. On leaves, stems, and thorns, circular lesions become raised and blister-like, growing into white or yellow spongy pustules. These pustules then darken and thicken into a light tan to brown corky canker, which is rough to be touched.